Hello everyone. I'm gonna introduce this topic through this video. My topic is to enhance the photothermotherapy through the inhibition of the heat shock proteins by using the upconversion nanoparticles. My name is Gordon Huang, Huang Guanbu. And firstly, UCMP usually composed of absorber and emitters. In this case, YB is absorbers and ER and TMs are emitters. Just like how it's been named, the UCMP can uh, absorb a photon with lower energy through the absorbers and upconvert into either a photon with higher energy or heat. And the distribution of the emissions is adjustable by changing the elemental ratio you want to use. And the photothermotherapy, as known as PTT, usually use nanomaterials as a photothermal agents and irradiate with a laser to produce heat. Then we further use that heat to kill the cancer cells. So this is how this therapy works. And as I mentioned, the UCMP can produce heat as well. So it can be applied as a photothermal agent here. But the problem is when the temperature of the cells increase, it will produce a kind of proteins called HSP, heat shock proteins, to prevent the temperature of the cells from increase too much. So my project is to enhance the photothermotherapy by accompanied by the inhibition of the heat shock protein to enhance the therapeutic efficacy. And in the beginning, to get the UCMP, we use a system called Shankline in our lab. It's composed of two channels. One of them is a vacuum channel, and another one is a channel flow with a nitrogen. Then we can use this system to get an environment with a high purity of nitrogen. Then we add the chemicals we want. And after some stirring and heating process, some additional solvents, and keep heating, stirring, then cleaning, dry it. Then we can get the UCMP powders we want. After a part with the Shankly, we can try to shine the laser on our samples before we clean it, and to see if it shows a green fluorescence. It's like a standard way to testify the quality of the UCMP. Then after we clean the UCMP and we dissolve it into a cyclohexane, this is color. It appears a little muddy. Then we can take these samples to measure the photoluminescences. And this is a top view of the measuring machine. And we can see the emission of the UCMP distribute at peaks, three peaks, which is 520, 540, and 6050. And the combinations leads to the green fluorescence we see. Here I refer to a experiment method from this paper. They use a melanin coating UCMP as a sensing platform for antioxidant capacity assay. So I follow up with their melanin coating process. Briefly, they add a kind of HCl buffer into the USMP solution to create an alkaline conditions. Then they add a different amount of dopamine hydrochloride crystal. After some stirring process, then we add the hydrochloric acid to stop a reaction.
Good morning.
Good morning. And from the CAT image, we can see the from A to D, we can see the thickness of the melanin shells increase as the reaction time increase. And since the melanin shell has been formed, the electrons will transfer from the emitters to the melanin shells and through the photon-induced electron transfer (PET), and the green fluorescence will quench. So from the spectra, we can see the quench fluorescence increase as the reaction time increase, but the absorption's coverage increase as with the reaction times due to the nature of the melanin with its broadband absorptions in visible range. So my goal is to functionalize my UCMP with coating a melanin shell on it and load the inhibitor of the heat shock protein 70 PES on the melanin shells. And I want to use this system into the photothermotherapy. And when the temperature reaches a certain point, the bonding between the PES and melanin shells will break, and the PES will try to block the function of the heat shock proteins. Then we can enhance the therapeutic therapeutic uh, therapy effect therapeutic effect. And currently, I've been preparing on my procedure of a melanin coating. We can see the color of my samples turns darker gradually as the reaction time, which is consistent with the results from the reference data. And the quench fluorescences and the increased absorptions are also consistent with the reference data. And so my future work will be trying to optimize the coating process to get the best condition and then to load the PS on it, then apply it into a photothermotherapy in vitro or in vivo experiment. And in a finish, from this representation, we can see my sample shows green fluorescence under 90 nanometers excitations. And after coating the melanin on on my UCMP, the fluorescence will quench due to the PET, and the UV phase absorptions of the melanin coated UCMP will increase. My future work will be trying to get the photothermal image of the UCMP to verify the enhanced temperature upon excitation and try to load the inhibitor of the shock protein on the UCMP. Then, more than that, it might be the TM, SDA, EDEX characterization of my materials and some visual study or cancer cells death through the photothermal. And this is my representation.